Hi everyone, I'm Angie Laird from Florida International University and welcome to our OHBM 2021 educational course on neuroimaging meta-analyses. We have a great lineup of talks for you that are designed to explain meta-analysis methods, give some practical recommendations, and allow you to gain hands-on experience. But to get things started, I'll be giving you a brief introduction and some historical context, which hopefully will provide a clear foundation as you learn about current methods and resources. Here in 2021, our broader scientific community is rightfully focused on the current reproducibility crisis, and we're developing amazing and innovative tools and resources and best practices for ensuring that our work is robust, reproducible, and reliable. But looking back 30 years ago, even during the time that fMRI was being developed as an imaging modality, a few groups were early pioneers in looking at consensus across neuroimaging studies, comparing activation patterns observed across these studies to determine where there was agreement. And when there wasn't such agreement, such as in the case of the anterior cingulate cortex, they examined the experimental design across these studies, which led to these two proto-meta-analytic studies on the somatotopic mapping of the ACC. The first one that I'm aware of was published in 1993 by Tomas Paus and colleagues. And in that paper, they examined various functional PET experiments that examined hand responses, speech, and eye movements, and mapped the variability of the response types across the cingulate. This paper was followed by a similar effort in 1996. And so you can see just from here that we've all been interested in the mediofrontal cortex for quite a while now. Below that are some additional very early meta-analyses. Methodologically, the focus across these was simply to plot the coordinates across papers and qualitatively assess convergence in location. And so what I want to make really clear right now is that we knew from pretty much the very beginning that there are limitations to any single neuroimaging study, such as small sample sizes, particularly compared to other fields of cognitive and social sciences. Also, we need to acknowledge that, especially in the case of fMRI, we are dealing with an indirect measure of neuronal activity, which has reliability issues that are limited by biological, technical, and methodological confounds. In addition, our field is chock full of publications of isolated findings, and that's because neuroimaging is an expensive way to do research, and additional experiments for confirmation and extension still are pretty rare. We also are dealing with generalization of context-specific findings, and that's because so much of our literature is based on linking brain function and pathomechanisms based on observed differences between two different imaging conditions. And so all of that is meant to introduce a topic for this educational course, which is neuroimaging meta-analysis, which asks the question, where do the locations converge? And the first topic that we'll be talking about is coordinate-based meta-analysis, which is enabled by two common community standards. The first is that we spatially normalize our brains to a standard brain template, and when we report the results of any given statistical parametric brain image, such as what you see here, for each cluster or blob of activation, we can extract the centers of mass or the peak locations and report those locations in standard brain space as XYZ locations. And so as a result, any given peer-reviewed paper is likely to publish a picture of fMRI activation patterns that are the result of some task, along with an accompanying table of what we like to call peak coordinates, which summarize those patterns and give you a quantitative representation of the brain maps produced by the study. And our literature is chock full of these map and table pairings. You just do a quick dive into the literature, you'll find table after table. And from our early days, we have considered how these coordinates can be plotted on the same map, representing results observed across many studies. And this led to the development of algorithms and analysis strategies for formal, quantitative, and coordinate-based meta-analyses. And so one of the key things to keep in mind is that when I say meta-analysis, I don't mean the standard effect size meta-analysis in which results are pooled to determine if a statistically significant finding is observed across studies. This type of meta-analysis has a complex history with some questionable practices sprinkled throughout the literature. So it's important to stress here that what we're going to be talking about is a location effects meta-analysis, which instead of asking, is there a significant effect, asks, of the significant effects reported across the whole brain in a group of studies, where do they converge? In coordinate-based meta-analysis, we ask, where do the coordinates converge? And so there's a number of different coordinate-based meta-analysis methods available to researchers, and they all follow the same general procedure. So let's walk through that. We first take coordinates that are harvested from the literature, and we convolve the coordinates, or foci, that's another word there, with spatial kernel to produce study-specific modeled activation images. We then combine those modeled activation maps into a sample-wise map, and then we compare somehow to a null distribution to determine voxel-wise statistical significance. So that's a brief illustration of the method itself. Let's continue to do an early dive into the early history. Back in 1994, Peter Fox and Marty Waldorf from San Antonio published a paper called Integrating Human Brain Maps that discussed possible meta-analysis methods, including image fusion and model-based synthetic analyses, as well as potential computational platforms to support such analyses. This is the first time that we had an article in the literature that kind of said, hey, folks, shouldn't we possibly be comparing maps across studies in a collective sense? But it wasn't until 1997 that this group proposed a specific method termed functional volumes modeling. And this was an ROI-specific way to model functional areas across studies as spatial probability distributions. Then in 1998, the San Antonio group laid out an early strategy in a paper called Beyond the Single Study for why meta-analysis was important and how it could be helpful for generating new hypotheses. And then finally, in 2002, a computational framework was proposed. The Brain Map database was described as a computational and community resource that would be able to facilitate meta-analysis broadly. And so beyond the efforts of the San Antonio team, honestly, not much progress was made until two independent papers were published in 2002 that proposed essentially the same strategy for assessing convergence across studies. The first was published by Chen et al. In this paper from 2002. They used what they called the AGES, or Aggregated Gaussian Estimated Sources approach, to assess convergence across verbal working memory studies. The second paper of that year was the more widely known study by Turkletov et al., which proposed the activation likelihood estimation, or ALE method. Again, they're both very, very similar papers, so I enter algorithms, so I urge you to take a look at both of them. But I'll note that ALE was soon adopted by the Brain Map database. And so by 2005, Brain Map was providing tools and software packages to help users conduct coordinate-based meta-analyses using the ALE approach. Meanwhile, an effort led by Tor Wager resulted in the development of a similar coordinate-based approach called KDA, or Kernel Density Analysis, which was initially described in a 2003 publication called Valence, Gender, and Lateralization of Functional Brain Anatomy and Emotion. And later, this algorithm was extended and was now termed multi-level KDA, or MKDA, as the extension treated peaks as nested within studies. And so I emphasize the MKDA approach because this is the algorithm used by another popular database called Neurosynth, which is used also for coordinate-based meta-analyses. Okay, so beyond ALE and MKDA, there are several other coordinate-based meta-analysis approaches that are available to neuroimaging researchers, and these were really nicely summarized in a 2017 publication. And so I urge you to also take a look at algorithms like the seed-based demapping and the scale approach. And then something that we haven't talked about yet is that we also have the opportunity to pursue image-based meta-analysis. Various different image-based meta-analysis methods were really nicely summarized in a 2016 publication by Momet and Nichols. And I'll note here that while image-based meta-analysis is preferred, given the additional information in the sense that statistical maps offer us the opportunity to really investigate spatial specificity across meta-analysis results, the maps themselves have more information than simply tables of coordinates. But image-based meta-analyses are fairly rare, and this is because a hardworking researcher can dig through hundreds of papers and harvest coordinates from tables, but they can't magically gain access to original statistical maps. Those need to be acquired from the authors of the original studies. which is why at this point in the presentation, I like to highlight the fact that NeuroVault is another community resource. It archives images from studies. So if we are going to engage in more image-based meta-analysis across our community, we really need to have access to the actual images. So it's a real great idea to upload your images to NeuroVault as a manner of standard practice when you are conducting your analyses and drafting your manuscripts. Because image-based meta-analysis really doesn't have a future unless all of us adopt this little bit of data sharing into our publication pipelines. So in closing here, I'll note that there are multiple coordinate-based and image-based approaches available to a neuroimaging meta-analyst. And it can be confusing to have your options distributed across different platforms and databases and resources and written in different programming languages. So one of the things that we'll be highlighting in this educational session is our new Python-based package called NIMARE, a Neuroimaging Meta-Analysis Research Environment, which gives you a central location and a common interface to conduct your meta-analyses. This is an open source community-based effort that Taylor Salo will be demoing later this session. So we really look forward to that particular activity. That's it for me. Thanks so much for your time today. I hope you enjoy the rest of the course and all that OHBM 2021 has to offer. And I look forward to seeing your meta-analyses out there in the literature. Thank you.
Hi, my name is Katie Bottenhorn, and I am a graduate student at Florida International University. Today, I'm going to talk about conventional or manual neuroimaging meta-analyses, or as I've come to think about them, meta-analyses the hard way, but don't be alarmed. So here's my plan for today's talk. First, I'm going to give a brief recap of neuroimaging meta-analyses, which will be more thoroughly covered by other talks in this educational session. And then I'll give a brief explanation of why in the world you would want to do one by hand, and give you a short explanation of how to perform a neuroimaging meta-analysis. And then we'll talk about some shortcuts or tips for semi-automated neuroimaging meta-analyses. And finally, we'll talk about why it's important to share your neuroimaging results as statistical maps, but by then I expect you will already know the answer. So a brief recap. Another lecture in this course by Angela Laird will be covering the background and history of neuroimaging meta-analyses, so we'll only talk about the basics here. But neuroimaging meta-analyses are made possible by the way that we do and report neuroimaging statistical results. Usually, these results are presented as statistical parametric maps, with brain activation peaks reported as XYZ coordinates and sometimes images shared in a common stereotaxic reference space like MNI or TALRAG. And meta-analytic algorithms capitalize on these qualities of functional neuroimaging studies to synthesize across results from different studies. First, to assess convergent activation patterns across an experimental task or paradigm, to compare neural recruitment across cognitive systems, to compare activation patterns across subject groups, or to define regions of interest, or ROIs, for future research. So each of these endeavors becomes much more important when we consider how much of the research that we've done so far has been underpowered due to low sample sizes. And we can use meta-analytic techniques to leverage this existing research to find common patterns of brain activation and help further our understanding of the brain and how it works. So some examples of impactful or novel neuroimaging meta-analyses over the years include a study by Fan et al. In 2002 that elucidated some subsystems of emotional processing, followed up by Buell et al. In 2014, showing support for a role of cognitive control in emotion regulation. David Glahn and colleagues in 2005 showed the neural correlates of schizophrenia extend beyond hypofrontality, which pushed our knowledge at the time. Fox et al. Emphasized the default mode network is not solely responsible for mind wandering and spontaneous thought. So as we develop more and better tools for automating meta-analyses, we have to consider why you would want to do one manually. So first we have to compare and contrast to explain what I mean when I say manual and automated meta-analyses. So manual meta-analyses proceed with first a search of a publication database or an online library for publications to include in your meta-analysis. And then the human being or human beings performing the meta-analysis have to pull coordinates and metadata from these individual publications, which implies that they have to read them. In some cases, even email the authors for images. And then using these coordinates and or images extracted from each publication, run a meta-analysis. In automated meta-analyses, instead of searching an online library, you just search a Neuroimaging Meta-Analytic database, which includes the coordinates and or images already pulled from each publication. So you kind of get to skip a step. And then you can just extract these coordinates and or images from database and meta-analyze. Each of these approaches has its strengths and weaknesses. Manual meta-analyses allow greater access to potentially relevant publications, and they are not limited by the bounds of a database, so they can include much more information that is included in any one database. And they offer the opportunity for hands-on quality control integrated into the data curation process. Cons include they are really time-consuming, more time-consuming if the body of literature that you're assessing is larger, and they do include room for human error in the data curation process. On the other hand, pros of automated meta-analyses include easier access to coordinates and or images and metadata as they have already been extracted from publications. There are several large databases to choose from, each with their own pros and cons. And you can outsource quality control to the population of these databases. Cons include they're inherently limited in their metadata as no single database can include all of the information about every paper. And there is usually a trade-off in the size of the database or the number of publications included in the database and the amount of metadata and quality of the extracted metadata. So allow me to make the case for manual meta-analyses by digging a little bit deeper into some of the pros of manual meta-analyses. First being that there are fewer metadata limitations. So not all publication metadata is included in every database, as I mentioned before, but there are a few that are commonly included, including the task performed in the study, the cognitive construct that was studied by the study, and participant qualities, so things like age, diagnosis, handedness, etc. So if you were to do a meta-analysis using uncommon data or data that is not usually included in a meta-analytic database, that would be a really good case for a manual meta-analysis. Some examples of such analyses include meta-analyses using novel or uncommon task definitions, such as one that some colleagues and I performed and published in 2019 on naturalistic fMRI paradigms, which can include everything from virtual reality to video games to film watching and listening to music, etc. As well as one that our lab currently has in the works about the social paradigms as grouped by the NIH's research domain criteria. Another reason to perform a manual meta-analysis is that you might have some uncommon persistent qualities that might not be captured by many databases. An example of which is the neuroimaging of the menstrual cycle, which was just published as a systematic review and meta-analysis, as well as a meta-analysis of heart rate variability, which was published in 2012 by Thayer and colleagues. So here is an example of some novel or uncommon task definitions and groupings from our currently in progress meta-analysis of social paradigm groupings according to the research domain criteria. So here you can see that each of these groups of social processing have different fMRI tasks associated with them, and doing a meta-analysis manually allows us to capture the construct that is being assessed by each of these different studies, even if they don't include the name of the construct specifically, or they do not specifically use one of these tasks. So another reason to perform a manual meta-analysis is that you have greater access to publications, as every database can only contain a subset of the literature. And which subset that is depends on how the database is populated. And there are generally two broad strokes approaches to populating meta-analytic databases, including manually populated databases, such as BrainMap, which are often biased by the distribution of tasks and cognitive domains that they capture. This is because manually populated databases are often populated by individuals who are performing their own meta-analyses. So as someone performs meta-analysis, the papers included in that meta-analysis end up included in the database, and thus they don't necessarily represent the distribution of tasks across the neuroimaging literature. On the other hand, automatically populated databases such as Neurosynth can be biased by publication year, while an automatically populated database might not have the same sort of bias according to task or cognitive domain. They're not necessarily continually updated, so they might lag behind the current research a little bit. And automatically populated databases are usually populated by some sort of text scraping algorithm that reads through through papers and extracts relevant metadata and coordinates or images. So in this greater access to publications, performing a manual meta-analysis allows you to more thoroughly comb the literature, which, like I mentioned earlier, can be helpful for less commonly indexed tasks, task groupings, or cognitive domains, and it includes the most recent publications. So here's a quick example. If we were to search Neurosynth for video games, you get a total of 0 entries. However, if you were to search PubMed for video game or video games, and fMRI or functional magnetic resonance imaging, you get a total of 298 responses. So that's more. And so you can see that in some cases, manual meta-analyses provide a greater access to information. So now that I've got you convinced that neuroimaging meta-analyses are worth doing by hand, we'll go through the steps of a neuroimaging meta-analysis, which are more detailed in more depth by Muller et al. In 2018 in a 10 simple rules for performing a neuroimaging meta-analysis. So we start by deciding on some inclusion and exclusion criteria, which are usually done when you define the scope of your meta-analysis and think about what question it is that you are trying to answer. Then you will go ahead and search for publications to include in your meta-analysis, read through these publications and annotate them with information that is useful for you in the process of performing this meta-analysis, and decide which papers to exclude based on your criteria, and then extract coordinates and metadata from these papers, and finally use that data to perform your meta-analysis. So here's a little example. What if we wanted to do a neuroimaging meta-analysis of video games in fMRI? Guess we'll find out. First, we're going to go ahead and decide on some criteria. There are some fundamentals that are kind of almost inherent to functional neuroimaging meta-analyses. The first of which is that the paper includes brain activation coordinates are reported or images that are shared in a standard stereotaxic space. And the second of which is that the paper includes whole brain analyses of brain activation. So for instance, not small volume corrected or region of interest analyses, which can bias your results. And almost everything else just depends on your use case and the neuroimaging meta-analytic algorithm that you're planning to use. So back to our example of video games in the scanner. Our inclusion criteria would broadly be that participants play video games as they lie in the scanner for. Exclusion criteria include coordinates are not reported or there is no access to images. Results are reported but not from neuroimaging data acquired during the video game paradigm, and results reported from a region of interest analysis, or not whole brain. So then you would go ahead and use these criteria to go ahead and search for some publications. There are some search engine options that are better than others. PubMed, Medline, and Web of Science offer really great options for searching the existing literature in a systematic way. Some less good but reasonable search options include Google Scholar and individual journals, tables of contents. And so basically what you're looking for when you're looking for a good option for performing a literature search for a neuroimaging meta-analysis is something that will allow you to systematically go through the literature and search it usually using Boolean search terms, so the and, not, and or, and nesting terms within it. So many meta-analytic researchers, in addition to searching these different citation indices and libraries, they will go through published reviews and the reference list from other publications to find more papers. So lots of ways to find information for neuroimaging meta-analysis. And again, returning to our example of video games in the scanner, a PubMed search to find information or to find previously published papers on this topic might look like this. So video game or video game no space, and fMRI or functional magnetic resonance imaging. And that'll cast a broad net and find all of the papers in PubMed that mention video game with or without a space and fMRI. So next you'll go through and read all of the papers returned by your search and decide which ones to include and which ones to exclude. And not all the papers returned by your search criteria or by your search will meet your criteria. So you'll have to go through and weed out the ones that don't. And a handy way to do this is outlined by the preferred reporting items for systematic review and meta-analysis, or PRISMA checklist, which can help you not only perform your search, but it can also help you decide how to report the results of your search. So I find it helpful, and others have found it helpful in the past, to review papers in stages. And of course, as you do so, you can keep track of your reason for exclusion. So depending on your criteria, it might be handy to first read just the abstract of each paper to get a feel for whether or not it meets your criteria. Or in the case of video games and neuroimaging, it might be more helpful to read through the methods to see if they in fact did perform neuroimaging scanner. So here is an example of a PRISMA reporting chart from that paper that I mentioned on naturalistic paradigms in fMRI. You can see that we started with 754 papers, and then we reviewed the method section to make sure that the stimuli were in fact naturalistic, and excluded way more than half of our initial search. And then we went through and more specifically reviewed the method section to see how they analyzed their data, and then reviewed the results of each statistical contrast to see if they included all of the coordinates. And here's a bit of a more thorough walkthrough from that paper that I mentioned on the research domain criteria social constructs. And you can see that it's very easy to start off with lots and lots of papers and then end up with a much more manageable corpus. So the next step would be to acquire your data. And in a coordinate-based meta-analysis, you will do that by extracting the coordinates from tables in the publication, and then separate those coordinates by statistical analysis, or contrast, a more commonly used term. And what coordinate-based meta-analysis do is they take the coordinates, which is considered the minimum viable information for reproducing or reanalyzing results. And many common algorithms attempt to reconstruct the statistical map from these coordinates by assuming that the coordinates are the center or focus of a more diffuse activation cluster. These assumptions are not necessary when you perform an image-based meta-analysis, which require you to either fetch images from a repository. So instead of using just the coordinates that are the peaks of a statistical parametric image, you can use the image itself, which provides a lot more information. And if the images are not available in an accessible repository, you can email the authors to request those statistical images, although I must caution that your mileage may vary in this case. And then the next step is to meta-analyze. Now that you've acquired your data, you can use one of the available meta-analytic algorithms. And this step is almost agnostic to the method of data acquisition or curation, as long as you meet the minimum requirements for the algorithm and software that you use to run it. Basic requirements include either coordinate or image data, depending on whether you're doing a coordinate-based or image-based meta-analysis, and any necessary metadata for the algorithm or software tool. So for example, the number of subjects is required metadata for most coordinate-based meta-analysis approaches. And then, of course, you need your meta-analytic algorithm. And as you will see later in this educational session, many of them are implemented in Nightmare, and here you can see that we have both coordinate-based meta-analysis algorithms and image-based meta-analysis algorithms. So all well suited to your needs. So that seems like a lot of work, reading through the literature, extracting coordinates and metadata, and then performing a meta-analysis. But what if there were shortcuts? I'm pleased to announce that there are shortcuts. And these come in the form of the semi-automated meta-analysis. So going through those steps again, if you're performing a manual meta-analysis, you have to decide on your inclusion and exclusion criteria, search for publications, annotate and exclude publications returned by your search, extract coordinates and metadata, and finally meta-analyze. So there are a few tools that can be inserted in this process that can speed things up considerably. The first is NeuroVault, which is an online repository of statistic parametric maps from neuroimaging publications. So if you are performing an image-based meta-analysis, you can find a lot of data that has already been extracted and curated there. So that kind of skips the step of having to extract coordinates or images in metadata by doing it for you. Another tool is Metacurious, which is a web-based tool for dataset curation. So you can go ahead and speed up step 3 by using Metacurious. And Neurosynth, which is an online database of coordinate-based meta-analytic neuroimaging results, or neuroimaging results for meta-analyses. So that can really speed up step 4. So Neurosynth is a coordinate-based repository that can help with searching for publications, so you can identify papers that match your criteria in Neurosynth. And then the coordinates and metadata have already been extracted, so you can go ahead and use Neurosynth to wipe out steps 2 and 3. And then there's NeuroVault, which is an image-based repository, like I mentioned, that can help with searching for publications and extracting coordinates and metadata. And finally, Metacurious, which is a web-based dataset curation tool. It is currently a work in progress, so it's not fully functional, but it will be in the not-too-distant future. It includes data from Neurosynth, but it can additionally help with searching for publications by querying the Neurosynth database and annotating and deciding whether or not to exclude papers based on the information contained in them. And finally, it can help with extracting coordinates and metadata as they are already extracted by Neurosynth. Which brings me to my very last point, public service announcement to all who are listening. Share your statistical results as images for people to find on the internet. It makes it much easier to perform meta-analysis, and it makes meta-analyses much more robust by using the full image data instead of just the reported coordinates. And it is further important to share not only your statistical like T or Z maps, but to also share your covariance maps, which can be used by many image-based meta-analytic algorithms for a more thorough assessment of prior literature. And that's all I've got. So thanks so much for your attention. And here are the papers referenced in this presentation.
Hello, I'm pleased to give you the next talk in the Neuroimaging Meta-Analysis course, a talk on a neuroimaging meta-analytic ecosystem. So more so than other talks, this is going to be a bit aspirational, talking about how things are right now and how we would like them to be going forward. So to do this, I'll review the existing tools that are available for intensity-based and coordinate-based meta-analysis, talk about some of the standards that are there for conveying the results of neuroimaging analyses, and then a proposal for an ecosystem. So a lot of these tools you may not know about for intensity-based meta-analysis. Intensity-based meta-analysis remains perhaps less common than coordinate-based just because there's been less data available. But it turns out for a traditional random effects meta-analysis, you can actually conduct these analyses in the existing software, whether that's in FSL's Feat, which is basically already designed to combine results of estimates and their standard errors, called COPES and VAR-COPES in FSL speak. There's a little known tool in SPM called SPM-MFX that does this. AFNI too can do this, and NIMAIR as well. You can do fixed effects meta-analyses as well in some of these packages. And then there are a number of other ways of combining images that are perhaps maybe less well known, but are standard meta-analysis tools, including Stauffer's, Fisher's, Stauffer's weighted. Some of these you can do just naively in regular neuroimaging meta-analysis software. Some of them need specialized software and have been implemented in NIMAIR. In coordinate-based meta-analysis, you've learned about different approaches. Perhaps the 2 or 3 most likely are listed here at the top: activation likelihood estimation, MKDA, and also sign difference mapping. So these have implementations that are available. Provided links here in the slides. You can get these and link through to them. And ginger ale is a tool that does some of these things, as does MKDA. And NIMARE is trying to be sort of a one-stop shop for all different types of meta-analysis tools. Down at the bottom also, there's a connectivity approach, meta-analytic connectivity mapping. That you can do in ginger ale as well as NIMARE. Now, what are some tools that actually I wish were more developed, and actually I think need more work? I'll just kind of give 2 sort of vignettes. One is in sensitivity analysis. In MKDA, the data from each study takes the form of a 0-1 map, which means the group analyses look something like logistic regression. And in logistic regression, there actually are well-developed tools for assessing whether there is bias in a meta-analysis setting. And these amount to saying, does the effect size go to 0 as the sample size goes to 0? And so you can ask questions like that. And so you can see examples here where this probably suggests there's some reporting bias, because we're having, there isn't much variation in the proportion of studies that are showing a response with a total effect size. And likewise here, this is sort of interesting. This is saying that the study size, the smallest samples are the most likely to have something active. And that's curious. So there's some work on here. This is from a paper by Tor Wager and colleagues. Another work that we just finally got into the literature was looking at the question of the file drawer. Would it be possible to estimate the number of studies that are actually conducted but then not actually published? This is something that a traditional meta-analysis outside of neuroimaging is something that people consider, but it's just harder to evaluate here. And we looked at ways of just using the total number of counts in a coordinate-based meta-analysis data set to see whether there's evidence. And we found that something like there's evidence for maybe in 1 in 10 studies that are basically 0, that there should be 0 foci studies, empty foci studies that should be in the literature based on the statistical characteristics of the counts and that we can see. So those are just 2 examples of robustness, sensitivity analysis, which I think could be better developed in neuroimaging. So if we want to sort of enrich the community of tools, we need standards for how we communicate the results from the neuroimaging neuroimaging software into the meta-analysis tools. So what am I talking about? Well, it's first what I'm not talking about. So I'm not talking about NIfTY. NIfTY, of course, is just a container for any kind of 3D data. I'm not talking about the software specific elements. It is true, there is a rich amount of data and metadata contained in each individual software's files, but they're not necessarily well described, not documented, and they're not sort of the result of a widely distributed community effort. Now, the BID standard, of course, is an exciting development that has been really transformative for neuroimaging, but the original BID standard was just for raw data before anything was done to it. And it's been very exciting. Just recently, we've had the BID's derivatives update, but when you look at that, you'll see that most of that is focused on pre-processing, smoothing, cleanup, application of atlases. It is not really telling you much on how to characterize the output of statistical models fit to neuroimaging data. So one effort that has tried to take this up squarely is the neuroimaging data model. So this is a beginning to end plan to use semantic web techniques to capture all of the metadata associated with neuroimaging study and analysis. So in the big picture, what it is that there are basically 3 stages. It's that the neuroimaging data model conceives of the study in 3 stages, comprised of an experiment, basically all the data, the details about the study before you actually collect any data, and basics of acquisition. Then there's the workflow, basically how you do the pre-processing and sort of pre-statistical analyses. And then there is the NIDM result, which captures all of the output that basically forms what of the results that get into a paper at the end. Now, the emphasis of this group that has been developing this was to initially focus on the results end. This is the thing that is relatively consistent in task-based fMRI, and this was built up. And the actual sort of heavy lifting of this project has been to develop the semantic web representation of all the details associated with a task-based fMRI analysis and the output of any particular software. And this is, frankly, a quite painful process, but at least this part of the model has been completed. It represents every element of sort of the design matrix, decisions made about the error, the techniques used for parameter estimation, what are the types of things that come out of that process, the different parameter maps and the residual maps. Everything is captured here. So this is very painful, but it now exists as a standard so that we can actually get an export from FSL or from SPM that captures all of the results in a standardized way. So all the sort of key and sometimes boring details about a statistical analysis can be represented in a machine readable format. And in fact, NeuroVault can actually accept and read in these files. But this is limited. This is just for neuroimaging task fMRI. And it isn't sort of just, it's just one piece of the puzzle. So led by Tali Arcani and with effort from Taylor Salo and Salo and others, we have proposed sort of a way to connect all the different pieces of a neuroimaging sort of analysis ecosystem to try to basically ensure that everything can be done with a machine readable manner to minimize error and to minimize effort. That's the idea. So let me step through this whole sort of idea for an ecosystem step by step. So we have primary analyses. So we definitely don't want to be in a place where we sort of say everyone has to use one tool. We want to be in a place where everyone is doing lots of different things and they all have different tools, and that's fine. But there's a common way to then actually export those results and feed them on into a curated data management system. And the current proposal is that we would use the NIDM results, since that is sort of mature and defined. And then there are also maybe some people who roll their own software. Now, the idea was that we have plans to help individual users make their own NIDM results packages, but this would ideally be, if not through there, directly uploaded to NeuroVault. So image-based results would go into NeuroVault. And then NeuroVault would coordinate with the central hub, which is creatively named NeuroStuff. So this would be a centralized place for image-based data as well as coordinate extraction. So using some sort of automated coordinate extraction and coordinating with other tools, using either code from Neurosynth or coordinating with efforts from NeuroQuery and other projects that you'll learn about, bring all of this with coordinate-based and intensity-based into one centralized place for images and coordinates. Now, that is just a bucket in a way, and the more annotated this storage is, the better. So for example, using the NIDM results standard is a way to make sure there's as much metadata as possible conveyed from the analysis software. But for historical results and results that were uploaded without NIDM, there may be a lot of information that's not specified. And hence, our proposal is to use an existing project, BrainSpell, to annotate and mark up these individual results and studies. There's another complementary project called Meta-Curious, which basically interfaces with BrainSpell to curate meta-analyses, and that too could interface with NeuroStuff. So from that central repository, the NeuroStuff repository, when you want to do a meta-analysis, we propose a NIMADS data standard. So this is a way to represent all the data you would like to feed into a meta-analysis. And this could be pulled off and queried through a library directly from NeuroStuff, but of course also could be that individual users may want to do their own meta-analyses, which is fine, but the idea is that this would be embedded in a NIMADS data structure with the help of a NIMADS Python library. And then once we're there, we're in a place where we actually can then do meta-analyses in NIMARE, which again, NIMARE is trying to implement all the widely used meta-analysis packages so that you basically don't have to choose. You don't have to any tool that you like, you'll be able to find in there and should operate as you're used to it. And so this can run, in addition to the number of analyses, you might want to interface to other things like a cognitive atlas to identify terms or regions, and you can do all that through NIMARE. Now the output of NIMARE then is proposed to then be sent back to NeuroVault, or you might be someone who just wants to do meta-analyses in NeuroSynth. Well, the plan is that for NIMARE to be the back end to NeuroSynth, but likewise, those results would be available in NeuroVault. So now this is focused, of course, on the project that Taylor and Tal have been working on, NIMARE. But again, as long as these have been all defined in open standards, it should be possible for anyone else to come in and drop in an alternative analysis framework and be able to communicate the results, take in NIMAD's specification for a meta-analysis, and then output results in a standard way. It is worth mentioning other efforts in this area. Simon Eichhoff's group has proposed ANIMA, an archive of neuroimaging meta-analyses. I think this started with 20 or 30. It's now got 61 meta-analyses. So it has an upload interface, and they do have an XML specification. Not sure if it's completely documented, but you can download all the details of each study in a machine readable format, XML. So this is good, and this is something I think is what has sort of been partly an inspiration to us, but it is not enmeshed in a sort of a workflow and a full sort of beginning to end and open collaborative environment, and that's what's been motivating our proposal for this particular ecosystem. So that brings me to the conclusions of this short talk. We believe that neuroimaging meta-analysis needs an integrated ecosystem. Everything from the primary analysis export, getting all the details, key details out of a neuroimaging analysis in a common fashion, a common way and place to archive all the output from study data, whether that's images or coordinates from historical studies, some way to curate that data and be able to update it when necessary or add more information as it becomes available. And then tools for selecting studies to actually go in and complete meta-analyses, and then actually conducting those meta-analyses and then interrogating them and reporting them out, and finally archiving those results. So this ecosystem that we've put forward here has been motivated to try to minimize effort and chance for error and maximize the reusability of all the data that goes into and comes out of neuroimaging meta-analyses. So I'll stop there and thank you for your attention.
Hello everybody, I am Jérôme Dockes. I work at the Origami Lab at the MNI, and I will talk about large-scale and automated meta-analysis. First, a very quick recap of manual, more traditional meta-analysis that have already been discussed in this session. So a broad overview of the procedure is that if we want to do a meta-analysis, for example about reading, we start by collecting many studies that use a reading task, and we extract the peak activation coordinates, the stereotactic coordinates that they report. We aggregate all of these coordinates and we smooth the result to produce a meta-analytic map. And then we can compare this meta-analytic density map to some reference distribution to threshold it and find the regions where activations are reported consistently in all of these studies and where we see a significantly high density of peak activations. And usually this is done by sampling many sets of coordinates from a uniform distribution across the gray matter and building like this, putting each of these sets through this pipeline and building like this for each voxel a histogram of the values we might expect under the null distribution of coordinates uniformly distributed across the gray matter. And then we can compare the true meta-analytic map to this histogram to threshold it and obtain our meta-analytic results. That's a very effective method, and I just gave a very approximate overview, but there are many variants. But it can be difficult to implement. First, because selecting the studies is subjective and very difficult and takes a lot of time. Also, the coordinate extraction is time-consuming and error-prone. And also because we only consider the set of studies that are about our topic of interest, for example, reading, and that we chose to include in the meta-analysis, and the rest of the literature is not taken into account. So we contrast the density in our group of studies against a uniform distribution, and this is very expensive computationally, because we have to do this non-parametric test and sample many set of synthetic coordinates, which takes a lot of time. And also, it's not a very realistic null hypothesis. So here, for example, we see the average density of reported peak coordinates across 13,000 studies about many different topics. And we see that coordinates tend to accumulate in some specific regions and are not uniformly distributed across the gray matter, regardless of the topic of study. So if we contrast our meta-analytic map to a uniform distribution, we might find regions in which coordinates accumulate regardless of the topic of the study, and that are not specific of the mental function or the disorder in which we are interested. So our results might lack specificity. A project that was developed to be able to do these meta-analyses at a larger scale is BrainMap. Meta-analyses with BrainMap follow the same general procedure, but many users annotate studies with labels from a small taxonomy and extract and curate the peak activation coordinates and insert them in one central database. So that enables doing these meta-analyses at a larger scale, and the BrainMap database contains over 4,000 studies, and possibly with higher quality data as the coordinates are validated by different users. Also, since the studies are tagged with some from the taxonomy, the study selection can be done with the query on this database and become somewhat more explicit and objective. Still, it's limited in size because to insert a new study, someone has to spend time labeling it and extracting the coordinates, so it grows very slowly. The tagging of studies with a few terms from a discrete taxonomy results in a rather shallow description of these studies, which limits the range of questions that can be investigated with meta-analysis. And also, BrainMap is not an open resource, so not anyone can download the whole dataset and perform different kinds of meta-analysis on it. Then the next important project for larger scale meta-analysis was NeuroSynth, and it introduces both a new way of building the dataset and collecting studies and a new meta-analysis method. So NeuroSynth collects the data completely automatically. It queries PubMed to get abstracts of many neuroimaging studies, and then it gets the articles from the websites of the publishers. It downloads the HTML pages containing the articles, and it extracts the peak activation coordinates automatically from these web pages. So as it's completely automatic, it was able to build a much larger dataset than the BrainMap database or whatever existed before. And the NeuroSynth dataset contains over 14,000 studies and 500,000 peak activation locations. This dataset is shared online and can be downloaded from this repository. And for each study in the dataset, it provides the frequencies of occurrence of over 3,000 terms from a manually created vocabulary of terms related to brain function and anatomy. And NeuroSynth also introduces a new method. So as I said, in the standard meta-analysis method, we select a set of studies and only consider these studies. And we look at the density of peak activations in these studies. So for each voxel in the brain, we will basically look in our group of selected studies how many times it's active and how many times it isn't. With NeuroSynth, we also take into account the studies that do not investigate our topic of interest. So for example, if we do a meta-analysis about reading, we will divide the NeuroSynth dataset into two parts. Those studies that mention the term reading in their abstract above a certain frequency threshold, and the studies that do not mention reading above that frequency. And then we also look for each study for a given voxel I, for example, if this voxel is active or not, meaning if it's within 1 centimeter of a reported peak activation or not. And like this, we can contrast the proportion of studies that activate voxel I in the group of studies that mention reading versus the group of studies that do not mention reading. And here, for example, so we contrast these two groups rather than contrasting this density to some null distribution or a uniform density. So here we see that the proportion of studies that activate voxel I is the same of both groups, and we don't see an activation between the occurrence of reading and this voxel. But in some other hypothetical example, for example, we could have calculation and some voxel that has the same proportion of activated studies when they do mention calculation, but a different one in the rest of the dataset. And here we see a different behavior of this voxel depending on the occurrence of the term calculation. And therefore we can see that the activity of voxel I is not independent of the occurrence of the term calculation in the study's abstract. And this can be formalized and tested with a chi-square test. So NeuroSynth doesn't simply look at for a consistent accumulation report of activations in a particular region, but it tests whether the activity of a given voxel is independent from the occurrence of the term of interest or not. And this means that we get more specific results, and as we only need to do a chi-square test for each voxel, we avoid the expensive non-parametric test, so it's also much faster. And finally, the studies are divided based on the frequency of occurrence of the term of interest, so there is no manual or subjective labeling of studies. And as we can see here, for terms that occur frequently enough in the literature, we can, like this, very easily obtain specific and high-quality meta-analytic maps. There is also an online platform where we can search for our term of interest and obtain in a few seconds a meta-analytic map. So this also changed the way that meta-analysis can be used, because before it was a lot of work and it took a long time to obtain a map. Now with NeuroSynth we can obtain one in a few seconds, so whenever we have a doubt, we want help interpreting an fMRI map, or we want to define an ROI, we can rely on meta-analysis across thousands of neuroimaging studies. Still, there remain some challenges with NeuroSynth, and one of them is that we need to select a single term that occurs frequently enough that we can split the NeuroSynth dataset in two. For example, reading occurs frequently enough that we can build a set of studies that mention reading and a set of studies that don't, and contrast these two groups. But if we have a more complex query that contains several terms, like the name of this contrast from the individual brain charting dataset, it might be more difficult to select a single frequent term and classify the NeuroSynth studies based on this term's frequency. Maybe in this simple example we could select one single term, but if we had an even more complex query, for example, a whole abstract or a text describing a protocol, we would need some multivariate way to combine all these terms and weigh them to build a meta-analytic map that takes into account all the components of this complex query. And we cannot do this with NeuroSynth. And if we wanted to use standard meta-analysis, we could, for example, try to look for studies that contain all of these terms. But as we add more terms, the number of studies that match all of them decreases rapidly. And we see that we cannot find enough studies to obtain meta-analytic results. So the new NeuroQuery tool was introduced to address this. And it's a multivariate model, so it will combine all the terms to produce a meta-analytic map. And we can see here the contours of the NeuroQuery output, and behind it the actual group level contrast from the individual brain charting dataset. Besides the new method that I will describe in a minute, NeuroQuery also comes with a new dataset that contains a similar number of studies as NeuroSynth, but it contains the full text of these studies, whereas NeuroSynth only contains the abstract. So in total, the NeuroQuery dataset contains around 20 times more text than NeuroSynth, and this means that a given term, here each dot corresponds to a term, is seen many more times in many more studies in the NeuroQuery dataset than in the NeuroSynth dataset, and that makes it easier to map these terms to brain regions. Then, as I said, the NeuroQuery tool has a very different model and approach to meta-analysis. So unlike NeuroSynth or more standard methods, it does not perform an in-sample inference. It does not perform a statistical test. Instead, it's a multivariate supervised learning model that maps high-dimensional representations of text to densities in each voxel in the brain. So it's a supervised regression model. The input features are term frequencies representing text, and the targets are activation levels of voxels in the brain. So to build the input features given text, NeuroQuery has a fixed vocabulary of over 7,000 terms, and for each of these terms, it measures its frequency, the number of times it occurs in the text. And like this, it builds a high-dimensional term frequency vector, and that is the vector of descriptors or regressors. These are the features of used by the regression model. And to build the targets, it extracts the peak activation coordinates and smooths them to obtain a density map. And like this, it obtains the density at each voxel, and each voxel is an output, is a regression target. So the activity of each voxel across studies is regressed on these term frequency descriptors, as we can see here. So if we look at the whole data set, the predicted intensities at each voxel is a matrix where each row corresponds to a study and each column represents the activity of a voxel across studies. The design matrix also has one row per study, and each column is the frequency of a given term in the NeuroQuery vocabulary across studies. And we regress these voxel activities on the term frequencies. So this looks a lot like the standard GLM used in fMRI, except that in the design matrix, each row doesn't correspond to a time point, but to study in the NeuroQuery dataset, and each column doesn't correspond to a condition, but to the frequency of occurrence of a given term. So again, it's a predictive multivariate model and not an in-sample inference tool. Also, it actually performs a reduced rank regression, and the details are not important, but what's important is that for rare terms, for example agnosia, they are not directly mapped by regression coefficients to brain regions. Instead, NeuroQuery relies on term similarities. It deems that terms that frequently co-occur in studies are related, and when it receives a term like agnosia that is too rare to be reliably mapped to brain region, it maps it to other related similar terms and relies on these terms to construct a brain map. So it's good to check the set of terms that it uses. So for example, here we have prosopagnosia, and here we see the weights of terms in the brain map, and we see that prosopagnosia doesn't participate. It's not included in the regression model. Instead, NeuroQuery finds terms that are related to prosopagnosia, such as face, fusiform, and so on, and combines all of these terms to predict a brain map. So it's always good to check this list of terms and make sure that if the term is not mapped directly, the proxies that NeuroQuery uses are a reasonable choice. And then as I said, an important feature of the NeuroQuery model is that it's a multivariate model. So we don't have to restrict our query to a single term, but we can input any amount of text and it will recognize and combine all of the terms present in the text because it's a multivariate model, and use all of them to build a prediction. Again, this is a prediction, not a map of test statistics on which we can threshold at some significance level. The dataset is also shared online and can be downloaded from this repository. And NeuroQuery is also implemented in a Python package, in an open source Python package. Finally, in the future, we hope to move away from coordinate-based meta-analysis used by NeuroSynth or NeuroQuery, and to use instead image-based meta-analysis using images in the NeuroVault repository. But at the moment, there is very little usable metadata on NeuroVault, so performing this image-based meta-analysis remains a challenge. An important thing to remember is that methods are not tied to a given dataset. So given, for example, the NIMR tool that implements many meta-analysis methods, we can apply any method to any dataset. So to conclude, a little summary of when to use what. If we have a set of studies that we have selected for a given topic and want to test their consistency, we can use a manual meta-analysis. To find regions associated with the text, we can use NeuroSynth. If we have a complex study or rare terms, we can use a predictive model that is NeuroQuery. And we have to remember that if we want to do reverse inference, that is, infer the role played by a brain structure, none of these methods will give us an answer, and we should turn to multivariate decoding with many output classes.
Hi, my name is Kendra. Today I'm going to talk about sources of error in neuroimaging meta-analyses. So I'll start with a quick review of some aspects of meta-analyses, and then for each step in a meta-analysis, I'll describe some selected sources of error and compare this error as it appears in manual versus automated meta-analyses, as well as in coordinate versus image-based meta-analyses, and explore potential solutions to that error. So first, the goals of meta-analyses include synthesizing past results, discovering new results, and generate hypotheses for new studies. The common steps in a meta-analysis are defining a question, finding the relevant literature, extracting data such as the reported results, analyzing that data, and then reporting the meta-analytic results. In neuroimaging, we have coordinate and image-based meta-analyses. Coordinate-based meta-analyses use the reported results coordinates from published papers, and image-based meta-analyses use the full results images that could be shared online, for example, in NeuroVault, which is a repository for such images. Conventionally, we've done manual meta-analyses where you manually search for data, go through exclusion criteria for each paper, and annotate the data that needs to be extracted, and then analyzing the data. However, more recently, we as a field have developed some automated meta-analyses such as NeuroSynth and NeuroQuery, and these just automate many of the steps in a manual meta-analysis, which allows them to work on larger data sets of papers. So first, we're going to start with the step of finding the literature and look at two sources of error in this step, publication bias and selection bias. So first, publication bias. This occurs when it's more likely for studies to be published if they have significant results. So in a recent preprint, Oudyk and Katherine showed this distribution of more than 1 million Z-scores from Medline, so from published papers in this Medline database. And you can see what could be the outline of a normal distribution, but there's this gap here in the distribution. Essentially, this is showing how there are fewer null results published than significant results. In coordinate-based analyses, we have the particular problem where if we get null results, there will be no reported coordinates. And so this raises the question of how do we actually include such null results in a meta-analysis? Even if we publish the null results, how would we use them? In automated meta-analysis, data that is shared from large databases like NeuroSynth and NeuroQuery for doing these meta-analyses don't actually include the statistics. They only include the coordinates of the results. So we can't evaluate publication bias, for example, in the way that I showed in the previous figure, looking at the distribution of Z-scores or other statistics. So we can't even evaluate publication bias. Potential solutions in meta-analyses are to evaluate and account for publication bias, and in individual studies to pre-register methods and publish null findings, including the unthresholded images, so that we can get a more realistic understanding of effects in our field. As a field, we need to value null results in order for people to be able to publish them. Next source of error I'll talk about is selection bias. So here you can imagine a population of papers that vary along some feature with the values represented by different fruits here. And say we selected these papers, you can see that we are not getting a representative sample of the whole population because we don't have any apples in our sample. So this is an illustration of selection bias. It's some reason that you're not getting a representative sample of the literature. So in an image-based meta-analysis, you have the problem that few studies share results images, and this is becoming more common, but perhaps some subfields or labs or collaborators tend to share more. And so if you do an image-based meta-analysis, you might be biased towards certain results from certain labs or subfields. Also, images and other data can be shared in various formats and locations. And so if you're only, say, looking for data in one location, you might not find all the data and thus get a selection bias. In a manual meta-analysis, you need to consider whether your search string is effective. This is a search string, a set of syntax and search terms that you use to find papers, say on a large online database of papers like PubMed. You also have to consider whether your database is comprehensive enough to to enable you to find all the relevant studies. So if you're looking on PubMed, for example, for working memory, would you expect to find all the relevant papers there, or would you also have to look at a more psychologically focused database such as another question is how objective are your exclusion criteria? So you don't want to have a selection bias, for example, worst case scenario caused by you only allowing papers into your meta-analysis if they show the kind of results that you're expecting. In automated meta-analyses, we have the problem that some cognitive processes or disorders or individual differences are better captured by terms that are included in the vocabulary that these systems work with. So for example, empathy could be a state, for example, a state someone's experiencing in a scanner for an fMRI study, but it could also be an individual difference in discussing trait empathy. So people might differ in how disposed they are to experiencing empathy. And if you just search for studies using the term empathy, you won't be able to distinguish these. So in meta-analyses, it's important to have objective searches in your manual meta-analyses and objective inclusion criteria. And it's really helpful to follow the established guidelines for doing so. And here we have the preferred reporting items for systematic reviews and meta-analyses, or PRISMA system, which gives you a template for reporting your search and your exclusion criteria. And I highly recommend you ask a librarian if you're doing a manual meta-analysis, because they have a lot of experience dealing with databases, probably particularly the subject librarian for your research area. And they have experience formulating the inclusion criteria for these kinds of studies. In individual studies, it's important to share results in standard formats and standard locations so that they're findable and usable. And as a field, we want to encourage best practices for publishing. So people doing a manual meta-analysis should include these kind of reports of their objective searches and inclusion criteria, and individual studies should share results images. Next step is extracting data, and here we'll talk about the problem of missing data, particularly when people do regions of interest analyses. So first, a whole brain analysis is done when you report results across the entire brain. So here we have a whole brain map, and we see the coordinates of peaks that might be reported in a paper. Alternatively, some people choose to focus on a certain area of the brain and only report results from these areas. And this makes it difficult to compare studies in a meta-analyses because essentially you're missing information from the rest of the brain. So something to think about when you're doing an image-based meta-analysis is, are the images whole brain maps? So did the authors post the unthresholded brain maps? And also during data collection, did they scan the entire brain? That's another reason why you might see not the whole brain in a shared image. In manual meta-analyses, this problem is simply addressed by excluding ROI meta-analyses, but we have the problem in automated meta-analyses that we can't distinguish automatically between regions of interest analyses and whole brain analyses. So all of them are included in the automated large-scale meta-analyses. Some potential solutions to this problem in meta-analyses are to exclude regions of interest results, and hopefully eventually we'll develop methods to automatically exclude ROI analyses in large-scale meta-analyses. In individual studies, it's important to report whole brain results and share whole brain results images where possible. So next we have analyzing data, and here I'll talk about how not all methods are equal in individual studies and how we should consider individual studies depending on the quality of their methods. And also we need to consider how methods influence results in the meta-analysis itself. So not all methods are equal. So one potential problem with individual studies that would be included in a meta-analysis is low power. So here we have a figure from a paper by Bottenhorn and colleagues showing the median power of studies included in neuroscience meta-analyses. So just compare this bar here where we have a power of 0 to 10% versus this bar here where we have studies powered with 90 to 100%. And I want you to ask yourself, should these individual papers be considered with equal weight in a meta-analysis, or should we put more weight on studies with higher power for which we can be more certain about their results? Another potential problem is ad hoc thresholds. For example, in fMRI, it was more common in the past, but it's still done where people include analyses with a very liberal height threshold and then an ad hoc, say, cluster level threshold. And in 2016, Auckland and colleagues showed that with certain software, for example, SPM here, and across different data sets, when you use such ad hoc thresholds, you can get family-wise error rates of up to like 85%, whereas you would ideally keep them around 5%. So studies that use this method will have more noisy results. And again, you could raise the question of whether such studies should be given equal weight in a meta-analysis if their results are less certain. We can also have other problems like software bugs that are discovered and corrected, and we have to think about whether to include studies done before the bugs were corrected, whether studies had adequate noise removal, and whether we have multiple studies within our meta-analysis that use the same data. This is becoming more common as people reuse large open data sets. So this is a problem in both coordinate and image-based meta-analyses, but particular for automated meta-analyses is the problem that we can't automatically identify papers' methods at this point. So we can't distinguish, more reliable results from less reliable results gathered with more liberal thresholds. Sometimes within a paper, people will report results using sort of conventional corrected thresholds, and then also include a more exploratory analysis with more liberal thresholds. And right now, in an automated meta-analysis, we can't distinguish those results. And further, even if you can annotate methods in a manual meta-analysis, some methods may not be reported. We don't know necessarily which methods are important, which are introducing more error and need to be accounted for, and we don't know how to account for them in most cases. In meta-analyses, one solution is to exclude very problematic analyses. One solution that we have come up with as a field is to downweight lower powered studies. And in the future, we can develop methods for accounting for other problematic methods. In individual studies, obviously you want to avoid problematic methods and share your data if possible. Even the subject level data is important to share so that, say, in a year or two, if we find a bug in the software that you are using, we can reanalyze your data and check that your results didn't change significantly after the bug was fixed. As a field, it's important to be aware of these problems when reviewing papers. For example, to catch papers that are using problematic methods before they're published so that they don't get included in the literature and in meta-analyses. Next, we have the problem that methods influence results. So just at a broad difference of methods is the coordinate-based meta-analyses versus image-based meta-analyses. And you can see here an example of each. And you can see how the results vary quite a bit between the coordinate-based and image-based meta-analysis shown here. And this is a problem across all these different kinds of meta-analyses we've been discussing. Potential solution in meta-analyses is to pre-register and plan your methods. And this is partly so that you won't be tempted to try various methods until you find the results that you expect. And it's important to report it if you do try various methods, and potentially also report consensus or a robustness analysis if you use several methods. As a field, we need to be aware of this when reviewing again. And finally, reporting results. And particularly, I'm going to talk about reverse inference as a source of errors in reporting the results of a meta-analysis. So first, let's distinguish between forward and reverse inference. So in forward inference, you start with a mental state. You look at the probability of brain activity given the mental state. So for example, you could make a claim like when people experience pain, we see activity in the anterior cingulate cortex. Whereas in reverse inference, you go the other way, as the name implies. You start with the brain and you look at the probability of a mental state given the brain activity. And you could make a claim like, when we see activity in the anterior cingulate cortex, the person is experiencing pain. And this, most of the time, is not a valid type of inference. There are special analyses that do valid reverse inference, but in most cases, especially in meta-analyses and what people use meta-analyses for, you can't make claims about the probability of mental states given brain activity. And this, again, is a problem across all of these methods. And the solution, the simple solution, is to just avoid inappropriate use of reverse inference. And looking forward, we might be able to do valid reverse inference if we have more large-scale open-ended decoding studies. So in conclusion, it's important to consider sources of error in meta-analyses, as in all research. I've been focusing a lot on errors. There are many good uses for meta-analyses, but it's important to be aware of these errors and mitigate them when possible. There are potential solutions for some problems. These are more established, like the PRISMA guidelines for developing exclusion criteria. And there are less established solutions which we could encourage, like sharing results images of individual studies. And finally, there's room for improvement. For example, for publication bias, we can work to develop more methods for evaluating and accounting for this problem. And that is it for this presentation, and I'm looking forward to talking with you in the panel discussion.